Welcome back, everyone, to the afternoon session of day 2 of the Breathomics conference. Thanks to all the speakers so far today, and Hugh for running the session this morning. So we will kick off the afternoon session with the first talk from Helen at Marks International, with the talk title, Advancing Breathomics Workflows, Innovation in Thermal Desorption to Support Large-Scale Studies. Over to you, Helen, and then we'll take some questions afterwards.
Hello, everyone. Thank you for joining my talk today. My name's Helen Martin. I'm the Business Unit Manager for Thermal Desorption Instruments here at Marks. Today, I want to introduce some recent innovations to help support scale-up of Breathomics workflows to large-scale studies. For those of you who aren't familiar with us at Marks, we're a specialist manufacturer of sampling, sample concentration, and automation and introduction techniques for volatile and semi-volatile chemical analysis, be that contaminants in soil, fragrance profiles, what's in your car smell, or the latest regulation for ozone-depleted substances in air. And of course, in collecting, concentrating, and analysing volatile organic compounds from human breath, skin, saliva, and so on for biomarker discovery and non-invasive diagnostics. Together with our sister company, Sepsolve Analytical, we form Scharnberg Analytics. Sepsolve complement Marks sampling and sample introduction techniques with their advanced separation technology with GC/GC products, detection and identification with time-of-flight mass spec, and suite of data analysis tools. My colleague Laura McGregor from Sepsolve has a fantastic poster showcasing solutions to support every challenge in biomarker discovery workflows, so please make sure to take a look if you haven't already. At Marks, our passion is solving challenges in volatile chemical measurement. We've been developing instruments and sampling accessories to support your analytical challenges for over 25 years. This workflow will be familiar to many people here today. Breath samples are typically collected onto solvent tubes via a bag or bespoke sampling device to collect and concentrate VOCs in breath and eliminate interferences like CO2 and much of the water. Tubes can be analysed straight away or stored or shipped back to a hub lab for future analysis. The accepted gold standard here is to connect TD with GC or GC/GC and mass spectrometry for analysis, followed by data processing, whether that be for targeted measurement of known markers or untargeted discovery studies. It's also worth noting here that TD is easily coupled with other analytical platforms like iMobility or PTRMS to bring the benefits of remote, in-clinic or in-field sampling and batch storage to different analytical platforms. Of course, that was a simplified view, and there are many, many more steps involved to prepare, collect and analyse samples. And it's so important to manage quality control at every stage, but also to balance the need of fast turnaround, ease of use, and of course managing costs. I could talk all day about the strategies enabled by thermal desorption to support the stages shown here, from conditioning, capping of tubes, automated tracking, humidity management, leak testing, and surrogate addition, and of course recollection of precious samples for archiving or orthogonal investigations. But what I wanted to focus on today is the considerations when scaling up to larger scale studies or clinical trials, something we've worked with many of you to understand and support over recent years. Automation will of course be vital. If you can't rely on automation, you're going to need to employ an army to manage the demands of such studies. Where are the opportunities to de-skill and enable non-specialists to perform as many steps as possible without too much training burden? But how do we balance those needs with embedding quality assurance throughout the cycle and ensuring our analytical system gives the same answer across hundreds of thousands of samples and studies which could span years? Preparation for sampling is probably the least glamorous stage in all of this, where it's easy to underestimate the importance and time required to prepare properly. You can insert your own favourite quote about failure to prepare and the resulting performance here. I have many of those. But a key area identified for improvement by many in this field is the instrument time taken up in verifying the background or blank of components in the sample chain, such as solvent tubes, masks, bags, mouthpieces, and so on. It's important to control the levels emitting from these so they don't contribute disproportionately to your ultimate results. And there's really no one-size-fits-all strategy here, and the proportion of tubes and samplers you choose to blank depends on your study design and ultimately what you're looking for. But it has become apparent that using the expensive, high-end GCMS that we use in our discovery and to process samples can cause really an unacceptable backlog in many labs when it's used to be blanking masks and solvent tubes ahead of the next sampling campaign. So to help solve this challenge, we developed our dual TD system. In this system, 2 samples are processed simultaneously on one GC, immediately doubling the throughput. Two TD100 instruments interface to a single GC with 2 columns and 2 detectors. The full system is controlled from one PC and one TD sequence table, so all the synchronization is taken care of for you. We can combine this doubled throughput with other time-saving features, like using overlap mode, in which tubes are desorbed to the focusing trap whilst the previous GC analysis is running. This means the trap is ready to inject to the GC as soon as it's called from the previous run, meaning the GC is kept continuously running samples and reducing the overall analytical cycle for every single sample. We also have the option to use hydrogen carrier gas, which can significantly reduce your runtime whilst maintaining good separation and sensitivity. Our collaborators at Alstone Medical put the dual TD to the test in their tube verification workflow, in which they test the background from sorbent tubes and medical devices before they're sent out for another round of sampling. Using dual TD, they were able to verify 440 samples in just 10 days, versus the 22 days it would have taken otherwise, a substantial time and cost saving. And of course, the Alstone team thoroughly validated the system to ensure they achieved comparable results from both channels, generating excellent metrics for sensitivity, accuracy, precision, linearity across both instruments running simultaneously. You can see data from the full study in a poster we presented together earlier this year, which can be downloaded from our websites. Now, another part of the workflow that's been repeatedly highlighted as a pain point by researchers in this field is managing the quality control burden whilst ensuring high fidelity data, particularly in longitudinal studies or high volume studies. How do we ensure our instruments give consistent results from the first sample to the 100,000th? Quality control strategies can be automated at many stages of a TD-GC-MS workflow, ensuring instruments are leak tight every time a new sample is loaded, as we've just seen, making sure background levels are below thresholds, and monitoring, to some degree correcting for, instrument response with a range of system suitability standards, quality control standards, internal standards, and surrogates. And ideally, biological quality control samples, which represent the compounds and concentrations found in typical samples and also include the matrix, not something easy to come by off the shelf for breath samples. So where can TD help here? Well, many of those steps can now be automated reliably, but some challenges remain. Loading standards and purging solvent is a delicate balance and can be time-consuming. Generation of biological replicates can take place at the point of sample collection, but this can be more costly in both collecting the sample and shipping and storage. And of course, for breath samples, we can't simply create a composite biological quality control by combining aliquots in a single vial. So to help overcome some of these challenges, we've introduced our Recollect 10 instrument. Recollect 10 is a standalone benchtop instrument which enables desorption of a single sample onto up to 10 TD tubes. This can streamline the loading of liquid standards onto tubes, with the added benefit of reducing the solvent, produce up to 10 replicates from a single sample, and if we do this for multiple samples and stack them together, we create a pooled replicate. And we can even stabilize samples collected with, say, swabs or thin film from the skin surface by transferring the analytes to sorbent tubes, so removing the risk of biological activity decay in the sample and extending its storage stability. So let's start with streamlining tube spiking. Manually spiking multiple tubes with liquid standards is time-consuming and laborious. Repeating this process for 10 tubes can take an hour or two. Recollect 10 enables you to spike one tube with a high concentration standard, then reliably split it over 10 recollection tubes, reducing the whole process to just 10 to 15 minutes. Importantly, the amount of material sent to each tube is 10% of what it was previously if you go to 10 tubes. This means that the solvent loading is also much lower, which reduces the purge time and lowers solvent consumption, all without compromising spiking precision. It also extends the analyte range, as it allows stronger solvents to be used as we're using less. The reproducibility of the splitting and recollection process used by Recollect 10 is demonstrated here by the near-perfect overlay of the 10 replicate TO17 standard tubes of 5 ppb, created from a single tube spiked with 50 ppb. We can apply the same process then to creating replicates from a single sample. Our collaborators at Loughborough University use a Recollect 10 to quickly and easily generate 25 composite biological samples. These samples were collected after peppermint oil ingestion following the peppermint protocol, to ensure each sample was different before they then generated a composite. On the left here, we have a reference sample washout showing the eucalyptal washout over 300 minutes, and then on the right, 3 example peppermint washout compounds which have been collected across that time series and then pooled to give replicate composite samples. These composite samples were then used for a storage study to enable Loughborough to easily compare the impact of their test storage conditions using samples that were actually the same and contained real biological matrix. This technique is also ideal for creating replicate biological samples for method development, for interlaboratory studies, or proficiency testing schemes, or to create a set of representative composites from across your study, which could then be used for further interrogation, perhaps of candidate markers to help identify them using different techniques, without you having to store a replicate for every single sample, which can require a lot of storage space and a great number of sorbent tubes. Which leads me on to creating pooled QC samples. In the same manner, we can create a set of replicates from original samples, which contain features from every sample, acting as a composite, for example, to intersperse through analytical sequences for quality control. Here we have pooled samples from 3 participants. The red trace shows the composites sitting in the middle of the original sample traces in blue, pink, and green. If I was to draw box plots from the same data sets using common breath features, we can see that many are remarkably consistent between people and some vary a lot more. No great surprise to anyone at this conference. And what we see with the points highlighted in purple, orange, and blue here is that the composite samples cluster towards the mean for each compound, a good proof of principle that our pooled sample represents the average of the individual samples and could be viable as a biological QC. To summarise, thermal desorption continues to evolve to support the challenging Breathomics workflows. It's our privilege and the thing that we most enjoy to work with you all and try and solve the real challenges that you have throughout the entire workflow, not just in the analysis. Strategies for ensuring data quality and sample integrity have been developed to support large-scale studies, and we continue to develop and work with you all on how we can best support these. And recollection offers more than just archiving. We can extend the dynamic range of our samples, create composite biological replicates, create pooled samples, and streamline and enhance standard loading workflows. And with that, I'd like to thank you very much for your attention, for joining my talk today. Thank you to Alstone for inviting me to speak, and thank you to our collaborators at Loughborough University, Matt Turner's group, and to Alstone Medical, Luke, Lauren, and Dean for their help with all the studies presented today. Thank you.
Fantastic. Thank you so much, Helen. We have time for a few quick questions. If you're able to unmute, can you hear us okay?
Yep, can hear you just fine.
Fantastic, fantastic. Thank you so much, Faris. There's so many exciting things that you guys have been developing that'll be so helpful to the breath community. So a few quick questions here. First one is, can any GC column and detector be used with the dual TD system?
Yes, they can. The important thing is that your method needs to be the same. Your oven method needs to be the same. You can't heat the columns at 2 different rates inside one GC oven. So you can use 2 different columns to, say, get parallel separations, or you can use the same to double up your throughput. You can use different detectors or the same detectors, but the crucial thing is the method needs to be the same.
Got it. Thank you. Next one is, can the Recollect 10 be used to create a calibration series? And if so, do I have to use all 10 positions?
So you don't have to use all 10 positions. You can use any combination of any positions that you like. In theory, it could be used to create a calibration range by splitting. So say you did 5, 50, and 500 milliminute. You could get a calibration range that way, but you wouldn't get too many orders because then you get into, say, 0.5 or 5,000 milliminute, which become a little bit impractical for good absorption. So yes, but in a limited way, I think. Brilliant,
thank you. Next question is, can the Recollect 10 be used to dry purge tubes?
Yes, it can. A real benefit of it would be if you wanted to add a standard at the same time. So you could dry purge 10 tubes while you're adding a standard to them. Say you want to add an internal standard for storage, for example. The best way to have more efficient dry purging there would be to use something like the TC20, where you can do 20 tubes at a time.
Yeah, we've got a lot of those. And then last question here is, will the Recollect 10 be reproducible for highly volatile compounds?
Yes, yeah, it will be. Again, it's kind of as per your sampling, really. If your sorbent's well matched and you're using the right flow rates to not get breakthrough, then the Recollect 10 will work in just the same way. In the TO17 data that we showed, we had things as volatile as propene and chloromethane, which are some of the most volatile things you'll trap on tube. So it works well for them as well.
Excellent. Well, thank you very much for joining for the questions, Helen. Thanks again for the presentation. And again, for anyone working in breath research, Marks is the place to go if you want to kit out your lab. Fantastic products, and I think those pooled samples will be really exciting for the breath field. So thanks again, Helen, for
Thank you
joining.
very much.